Then I'm going to share my profiling molecular interaction by using. Let me briefly introduce the functional protein microarray. So first is to express the protein in high throughput way. Later on is to print on the cover slip like 1 by 3 inches. And later on is to print immobilize on the cover slip to preserve the conformation of the protein for the large scale screening. So the screening can be involved like to decipher the protein-protein interactions and like a finding of homology to the downstream proteins, for example. And also can be profile the antibody binding, like the serum and the protein microarray as a antigen platform. So what kind of antibody should bind to the protein and which antigen should bind. And also the small molecule binding, such as drug screening or drug specificity test. And also like a substrate identification, including like identify the phosphorylation substrates for kinase or for E1. So and there's more application like finding the unit binding proteins or RNA regulatory protein, etc. So today I'm going to share 3 of my research projects. This is the first one. Because there's a pandemic of COVID-19. And before COVID-19, there's more coronavirus exist in the population. That's called common cold coronavirus. It includes like 229E, NL63, HKU1, OC43. That accounts like a 15% of common cold cases. So that means all of us might get this kind of coronavirus already your past life. And there's some SARS-CoV we test fatality rate 9%, and there's a related SARS-CoV to case fatality rate around 1 to 60%, 1 to 6%. And there's a rare case called MERS-CoV, death rate is 37%. And they all have a great similarity because they belong all to the coronavirus. So in order to know the pandemic, when the pandemic occurs, the immune response, whether it's specific to certain like a SARS-CoV-2 or not, that's a big question to ask. So we produce a protein microarray, include all the viruses, antigens that I mentioned, and put in the microarray. And it's because that is not much protein, so we print 16 identical blocks for different specimens. And each block have a good reproducibility with R squared 0.984. So means that's quite reliable for the detection. And later on, we want to test whether the sensitivity of this protein microarray is good enough. So we use zero dilution of the antibody and found the LOD is 19 picogram. It's like a 1,000 more sensitive than traditional ELISA or rapid test. And later on, we want to show the protein of the array have a function and the conformation is correct. So we use a post-human receptor called ACE2, and it shows dose-dependent binding, and the LOD is around 700 picogram. So this show the detection platform is quite reliable and sensitive. So later on, we want to show the immune specificity for the COVID-19 patient. So we label the human IgG and IgA, trying to profile them. This on the left-hand part is a COVID-19 patient. You can found they have good response to the IgG and also have a great response to the IgA. And some patients just only respond with IgG but no IgA at all. So there are some subject differences. And we actually find some good biomarkers with combination of IgG or IgA with the sensitivity around 97% and specificity around 97%. And after this, we found out almost all the 80 to 90% of Taiwan people already previously infected with the COVID-like coronavirus, and it generates some cross protection to the COVID-19. But it's a big difference to the Western world. They have less abundance of the antibody against the common cold coronavirus. So that will generate less protection against the COVID-19. So that's our previous publication. And I want to talk about the second project because the pandemic ongoing and there's a lot of mutation. So there's a alpha, beta, gamma, delta, omicron, and cause a lot of waves of the infection. And even for now, it caused a big wave due to the opening of the China. And there's all kinds of at least 3 kinds of the vaccine available, like Moderna, Pfizer, and there's AZ in Taiwan. And all this virus is meant to generate the protective antibody to prevent the infection happening. This kind of antibody called neutralizing antibody, it can bind to the virus and block the interaction to the host cells through the ACE2. So with this concept, we produce a so-called SARS-CoV-2 variant called variant protein microarray. It contains all the current variants spike protein on it. And because the protein on the array is functional, it can bind to the host receptor ACE2. So without any interference, all the variant can bind to the ACE2 labeled in red. And if there's an interference, like when you get an injection with the vaccine or you have antibody drugs, you will see the antibody bind to the spike protein labeled in green and prevent the red binding of the ACE2. So this is kind of competition assay between to mimic the infection. So by seeing green means more protective, seeing red is more pro-infection. So let's see what's the clinical test. We get some vaccinated subjects, and they are mostly showing green or red, greenish. And for the unvaccinated subjects, they are all showing in red. And with different vaccine capsules, we can see like AZ 1 dose, AZ 2 dose, Moderna 1 dose, Moderna 2 dose against wild type. We can see there's a big subject variation in the AZ vaccine. And if we further analyze, like in the AZ 2 dose, we can see the protection neutralizing antibody decrease due to the variants from wild type, alpha, beta, gamma to omicron. It only preserved like 20% of the neutralizing activity. So this is a single assay can differentiate all kinds of antibody and infection status in each subject. And we also can provide the specificity of the drugs. There are some famous ACE2 inhibitors labeled in here. And in the wild type, the inhibition curve have a dose response. When the drug is higher, it inhibit have a higher neutralizing activity. But when it comes to delta or delta plus, the inhibition is totally flat. The drug is lost its function due to the variants of the virus. So this is useful for the drug development. So this is my last project I want to share. It's called G14 couple receptor. Perhaps you guys already know. So GPCR is important in the drug development. So, however, there's challenges because GPCR is difficult to purify and have a 7 transmembrane domain. So we try to use the virus to carry the GPCR for us and bring the first of the kind GPCR protein microarray and published in Nature Communication. And we also provide for the antibody drugs, some is very good, specific, and also there's only half of the antibody drug is specific and half is failed. We can profile the specificity of the ligands, and we can find the infection receptor for the group B strep, which is called the neonatal meningitis. And we find the GPCR can bind to this bacteria, and we validate in vitro by inhibit the infection, and in vivo to block the meningitis. So this is the summary, and I want to call for some collaboration. First, we can provide some customized protein microarray with high content, and we can provide a low volume, high precision printing. We can print each spot 100 picoliter, very little amount of protein consumed. And we can profile the molecular interaction with labeled molecules. If you have any interesting molecule, you can provide me for the profiling. We can offer serum profiling for any patient, like an autoimmune patient or any rare disease patient. And what I'm looking for collaboration is some chemists for the surface treatment or some variety of 3D surface treatment or polymers. I'm looking for the label freeze detection like a nanowire, bio-FET, or some SPR technology, because sometimes labeling will damage the small molecule characteristics. And I'm looking for automation and drug screening. And at the last, I want to make an acknowledgement of my lab member and some collaborators all around the world. Thank you.